our pleasure to bring the session to you this afternoon around facial eczema. Presenting for us this afternoon, we have Esther Donkersloot. Esther is a quantitative geneticist by training and has completed a Master of Science in Animal Sciences at Wageningen University in the Netherlands. Esther came to New Zealand and LIC in 2014 for her Master of Science thesis and has since then graduated and worked in animal evaluation and the International Department of LIC, and is now a scientist in our research and development group and heavily involved in the Resilient Dairy Program. Presenting alongside Esther this afternoon, we have Steve Davis. Steve is employed as a senior scientist at Livestock Improvement, working in the genome biology team within research and development. A long time ago, Steve was a research scientist involved in various aspects of mammary gland biology at Ag Research, Ruakura in New Zealand. Up until 2011, he worked for Fonterra, the major milk processing company, identifying genes that alter milk composition. Current research is focused on the development of milk diagnostics for facial eczema, together with Lorna McNaughton. So this afternoon, Esther and Steve are going to talk to us about facial eczema. Esther will talk to us about how this fits into the Sustainable Food and Fibre Futures Resilient Dairy Program, and then Steve will focus on the nitty-gritty detail around facial eczema and will answer some of the commonly asked questions for us. So without any further ado, thank you, Esther and Steve. I will hand over to Esther.
Thanks very much for joining us this afternoon, and thanks, Joss, for your very kind introduction. So yeah, my name is Esther Donkersloot. Apologise for my Dutch accent. I think it's still a bit there, but we'll move on to Steve rather quickly. I just wanted to take the opportunity to very briefly talk about the Resilient Dairy Program and where today's talk fits into this. So as hopefully most of you are aware, LIC is leading the Resilient Dairy Innovative Breeding for a Sustainable Future Research Program, which is an investment and support from MPI and Dairy NZ. The 7-year Sustainable Food and Fibres Future Program, SFFF, was launched in June 2019 at the National Field Days. In short, I don't want to take up too much of your time, but just want to give you a brief overview. Resilient Dairy really seeks to utilise disease management technologies and genomic advances to enhance the health and well-being of the national dairy herd, the resilience of our cows in New Zealand. To capture this large program and the amount of work that falls underneath it, we describe it under the 2 different headers, as you can see on the slide. So on the left side there, we've got animal well-being and health diagnostics. And the first bullet point underneath that is disease and well-being screening. And this is all about developing diagnostic tools around providing early identification of diseases in our dairy population, for example, mastitis. And part of this work is really utilising some of the data and tests that you as farmers are already doing, like herd tests, and developing new techniques to identify animal health traits such as pregnancy status and yonis. The next one there is biosecurity screening. So that's all about developing diagnostic tools, again, to provide early identification of notifiable diseases. So this could be Mycoplasma bovis, etc., etc. And samples, again, could come from bulk milk, a herd test, effluent, or feces. So that's what we're working on that one. The next bullet point there is facial eczema diagnostics, which we will hear all about from Steve today. So I won't go into that one. Very quickly moving on to the other side. So we've got genetic innovations and genomic advancements. And the first one there is genomic inbreeding. And within that subtitle, we're working on really improving the accuracy of inbreeding in the animal evaluation model and including inbreeding into our breeding program to increase the long-term health of our dairy population. Next one is genomic modeling, again, all around the animal evaluation model and how we can incorporate new phenotypes and account for structural variants within the genome to increase the accuracy of the model and increase genetic gain for you as farmers out there. Next bullet point there is incorporation of the New Zealand specific genomic reference. This has been a massive piece of work that we've done a lot of work on already, and it's all around being able to update the reference genome, which was currently been held by American Hereford, to a New Zealand breed specific genomic reference. And then last bullet point here is, sorry, not the last one, second to last is facial eczema. And again, that's what we're talking about today. So I don't want to steal Steve's thunder on that one. And also another piece of work there is the Y chromosome investigation, which is all around determining the influence the Y chromosome has on key breeding areas such as male fertility. And last but not least is Dairy NZ's InfoHertz, and that's the investment from Dairy NZ around rebuilding the national evaluation system for dairy cattle to incorporate genomic information to facilitate faster rates of genomic gain. Yeah, that's Dairy NZ's piece of work. So before I hand over, I just wanted to share with you that there's a lot more information around resilient dairy on the LIC website under the heading research, and underneath there again the header resilient dairy. We also endeavour to utilise things like these chat chairs to communicate the pieces of work that we're working on back to you, shareholders and also interested parties. So without further ado, I want to hand it over to Steve.
Thanks, Esther. Hello, everyone. What I'm going to cover just in the next 10 minutes is really a very brief background on facial eczema, cover what some of the costs associated with facial eczema in dairy, how people are dealing with it, and then we'll cover off some of the newer diagnostic methods that we're trying to develop at LIC with a view to breeding for facial eczema resistance based on natural challenges with the facial eczema toxin. And that's the toxin there. That nasty little diagram at the bottom is the facial eczema toxin sporidesmin, which is found in the fungal spores, which the picture in the top right-hand corner there of the slide is an actual picture of the spores of the fungus. And that toxin, the key part of that toxin is the SS bit in the right-hand side, and that's what does all the damage, which basically causes liver damage in animals that are so affected.
Facial eczema is caused by injection of the toxin sporidesmin, which I just showed you the picture of, from the fungus Pithamyces chartarum, which grows basically on the leaf litter at the base of the pasture. It impacts ruminants, and that's really mostly to now has been in the North Island, but in bad years it certainly has been known to spread, particularly down the west coast of the South Island and around that sort of Nelson area. An ingestion of the spores with the toxin causes damage to the liver. Arguably, facial eczema is a misnomer in terms of a title for the disease. It's really about liver damage caused by the toxin. And the diagnostic up to now has been release of an enzyme from the liver. The damaged liver releases the enzyme into blood. The vet comes along and measures the activity of the enzyme in blood via Gribbles or similar organization. And that's basically diagnostic for the liver damage. And usually at the time of the year that facial eczema occurs, between January and April, that's associated mostly with ingestion of the toxin. And farmers traditionally recognize facial eczema as only in the clinically affected animals. So we see sloughing off of the skin, particularly around the udder and around the neck region, and inflammation of the teats and that sort of thing. But where there's clinical facial eczema, there's often a lot more subclinical, as I'll show you shortly. And that's a picture there of Emma Cutton's collaborator holding a nice healthy liver, and that's taken actually from a video that she does on the subject, showing you the difference between a good-looking liver and a not so good-looking liver. But when clinical facial eczema occurs with those external symptoms that I've just described and shown you, usually something like 50 to 100 times more cases of facial eczema than that are subclinical, so not showing those clinical symptoms. And those animals that are affected like that with relatively or somewhat milder liver damage still show considerable production loss. The clinical animals are really just the tip of the iceberg. There are a lot more animals that are affected when you see clinical cases of facial eczema. And the cost, this has come from the Dairy NZ website, where there's a cost calculator, which I'm pretty sure was developed by Emma Cuttons and Winston Mason, her collaborator at VetEnd. This is you can use this calculator if you put in whether you've got a low facial eczema, moderate facial eczema, or severe. Put in your cow numbers, and it'll compute the best guess at what that's costing you. So for a 400 cow herd with moderate facial eczema, the cost in terms of production loss is estimated to be about $6,500, and then there's other costs that can be associated on top of that, which come with decreased live weight gain or increased live weight loss, and also there can be deaths obviously associated with really severe facial eczema. So it is a significant cost overall, and the others have calculated sort of for the industry, it's a $50 to $100 million cost per annum. But of course, the major cost really is probably reputational damage to the industry around facial eczema, because it is a big, or potentially a big, welfare issue. And one of the problems that we've had with facial eczema, I think, over the years, and getting it recognized as a case deserving of really quite major investment in terms of research, is that it only crops up badly, probably every 6 or 7 years. And in between time, there can be groups or regions which are badly affected, but it's not really affecting the whole industry simultaneously, and therefore doesn't get the recognition it really deserves. But at significant cost, $50 to $100 million for the industry is a best guess. And in terms of some of the tools we've got at the moment, then I think most of you, certainly the farmers amongst you, will recognise, and the vets will recognise the spore count information that's published either on the Gribbles website or the Vitora website, and there's probably a bit more information out there as well. But the Gribbles site expresses the affected, or the spore counts, in terms of the percentage of counts that are more than 30,000 per gram of pasture. So the farmer has to go out and, or focus farms for this exercise, will go out, clip an area of pasture, and then wash that with water, then count the spores in the water and convert everything to spores per gram. And this illustrates, and 30,000 spores per gram is really the magic number. That's the threshold above which you run the increasing risk of seeing liver damage associated with the ingestion of the spores by the herd. And that's the data for the last 4 years. If we went back to 2016, then the percentage of counts going above 30,000 was above 70% in 2016. That was really the last really bad year. But you see 2020, last year, COVID-19 might have been rampant, facial eczema not so much, but we did see amongst them, and I'll show you in a minute, we did see among herds that we looked at quite a lot of facial eczema was around, but very localized. This year, spore counts went up, peaked about 3 or 4 weeks ago, dropped after we had some rain, and then climbed back up again when the warmer weather and the damp conditions we had today's Gribbles chart would see that coming down over the last few days, but I think probably in association with the rain. And the Vitora website is a little bit different in that it shows just straight spore count, the red line there being 30,000, and that's for the Otahonga district. The Vitora site is quite nice because it's granular in those districts, but you'll see there some of those counts getting up to 200,000 per gram, really starting to get quite high risk for the herds in those areas. Okay, so spore counting is a technique we've got. It's relatively simple, a little bit time consuming, but the data is out there, bearing in mind that farmers really need to know how their farm relates to what is being published by these sites. And dealing with facial eczema, now zinc prophylaxis is probably the most well-known way of trying to prevent the impact of the facial eczema toxin. But Emma's work, particularly over the last 5 or 6 years, has shown that 70% of farms are failing to provide enough zinc to the animal to provide the zinc protection. So in terms of the techniques that are used, then the zinc bolus is the most surefire method of getting zinc levels in the animal up to a high enough concentration to achieve prophylaxis against the toxin. The next one would either be probably drench or adding the zinc into the feed, which is not always practical for all farmers, but those farmers who do feed in the shed, that's probably the best route of getting zinc at the appropriate rate into animals. And then the last one there, which is the one that Emma has emphasized, is that putting zinc in the water supply really does not work as a means of getting zinc protection against facial eczema. And that's probably the simplest thing that farmers can do now, is change the method of provision of zinc to at least give the herd some protection against the toxin. And it's not a sort of universal panacea. If the load of toxin and spores is high enough, then it will overcome the protection of the zinc. So we've seen a farm this year that's been using the zinc bolus, but still managed to get hit fairly hard by facial eczema in spite of that protection. So it's not perfect, but it does raise the threshold at which the toxin can act. And the other point about zinc protection is it's not sustainable. So the Europeans now are starting to ban zinc in animal production systems, particularly in pigs. So there's probably only a limited window where we're going to be able to carry on using zinc for protection against facial eczema. Then breeding, the breeding for facial eczema resistant sheep, I think has been going on now probably for about 30 years. It's been going well. The level of protection amongst sheep in New Zealand has been increasing year on year as the genetics has been passed on into the industry. So that's good. And CRB offers their FE product based on bull testing. We don't really know how good that is at the moment, but it is based on the sheep system. And the other nice thing about the sheep work is that the genomics angle has been unraveled in relation to facial eczema resistance. So there is a prediction tool which is reasonably accurate, certainly as accurate as some of the milk testing genomics predictors that we've got. The ability to use genomics is something that we want to aim towards, which I'll come back to at the end of this talk. The third point is that pasture and crop species is particularly important in relation to development of the fungus and sporulation of the fungus. So summer crops in particular don't necessarily carry the same spore count as ryegrass. The fungus particularly likes ryegrass, so we can get away from ryegrass with crops like chicory and plantain and maybe maize silage. Getting away from ryegrass is good in terms of reducing the toxin load on animals. Tall fescue has a little bit of data around tall fescue does not promote the growth of the fungus. Same little bit of data around kikuyu, which is obviously relatively more prevalent up north. And then silage can be useful as well. So in terms of getting rid of the fungus and getting rid of the problem, for me personally, that's I think a favorite area to do something in the summer that gets you away from reliance on pasture, which obviously you can combine that with some drought insurance as well. There's double the advantage. And then there's other things that we can be used dealing with FE, like fungicides, which I understand I don't really know a great deal about it, but they're quite expensive and can be quite toxic. But the timing of fungicide treatment needs to be quite accurate relative to the growth of the fungus. And then grazing management before Christmas, really to try and limit the amount of leaf litter that the fungus can grow on is also important. I guess one of the questions that we asked a couple of years ago is, can we basically produce a milk-based test in that we're getting milk in for herd testing on a regular basis from farms? Can we use that milk as a herd test to detect FE exposure? And this was particularly perhaps aimed at getting a phenotype where we can understand the genetics a bit better. And would avoid then having to go out and bleed whole herds, which would be relatively expensive. And then following from that, can we develop a breeding value to help breed for more FE resistant animals? So the concept really has evolved from there to identify herds that are afflicted with facial eczema through a biomarker test on milk, and in this case it's going to be bulk milk. And then can we blood sample for gamma GT, because at the moment that's really the cheapest test we've got in blood that we can use to detect liver damage. And then over time, can we then use GGT to develop a breeding value, and with time a robust genomic prediction? Going back 20 odd years at AgResearch, and Chris Morris and Neil Cullen, still there, did demonstrate that you could develop a breeding value based on natural exposure to toxin and with relatively high heritability. So there is, if you like, proof of concept, at least that far along the line. So this is a diagram of some of our first results with a milk biomarker that we, looking at the dates there from basically from the middle of January to early May, and this in 2018. So it was really our first attempt at looking at change in the milk biomarker over time. So the normal level of this biomarker is in the range of sort of 50 to 65, 70. And going outside of that range, we know correlates pretty well with the GGT levels in blood. What we don't know quite clearly is as to when does that level of biomarker indicate, when's the threshold at which we can say with some surety that the animal is, or the herd is being quite badly affected with facial eczema. But we think, based on this year's data, which we're still collating, that it's somewhere in the region of 80 to 100 is really indicative of around 10% of the herd being moderately affected with facial eczema. So this is an example of one herd where from mid-March onwards, the levels of the biomarker increase up to over 120, which we think would be of the order of 30 to 40% of being moderately affected with facial eczema. And then cows get cold, dried off, etc., and the levels drop down quite quickly towards the end of April, and then rise again. Our expectation would have been then that the spore counts were still high and were causing liver damage in the animals that remained in the herd. And one key thing about facial eczema is that the toxin gives a cumulative response. So once you've got a bit of damage, the next dose of toxin adds to that damage and keeps adding. So things keep getting worse unless you can get the animals off the pasture. And then last year, which was an experiment that was really curtailed by COVID, so at around 9 weeks, we had to stop the experimentation. That was corresponded with us going to lockdown at the end of March last year. But these were sire proving scheme herds that we were following on a week-by-week basis from, I think, mid-January onwards. And you can see there that in the top group that there were some clear elevations in our biomarker, similar to the first graph I showed you. And the second block of farms below there, we've got biomarker levels going up to 160, 170, at least on one of the farms. And where we've managed to get bulk milk samples off farms where we know they've been absolutely smashed by facial eczema, 80% of the animals being affected, some deaths, then we're looking at biomarker concentrations there of 250 to 300. So really very high levels. So that's where we're at with the biomarker at the moment. And this year we've been going out and getting a lot more blood samples off herds that are showing relatively high biomarker concentrations and showing the relationship there between the incidence of facial eczema as measured by GGT and the level of biomarker in milk. So we think we can identify herds affected by FE well before, might be a bit extreme, but we certainly think we can get significantly before clinical signs become apparent. So we're able to identify herds that have lots of subclinical cows. And the big question there is, what can farmers do about it if they know that they do have that burgeoning problem within the herd? This year, rather than doing sort of 10 or 12 herds, which is what we've done up to now, we've got a much larger data set being collected and to firm up on those diagnostic levels, but it'll be of the order of 40 to 45 herds that we'll have data for this year, and about 30 of those will have gamma GT levels. And the nice thing about this year is we've done a lot of sire-proving scheme herds, which will allow us to tap into the genomics if we can get enough herds and animals being affected in those herds. So for the future, we think we've got a diagnostic test based on belt milk, which would be quite nice because it'll enable farmers really just to have to tick a box when the belt milk is belt milk will go to milk test and then get sent out for analysis. So there should be about a 3-day turnaround on a sample that potentially is being provided to milk test every other day at that time of the year. And we've got the potential for an individual cow level test based on the herd test, but we've got to get the cost of the test down, biomarker down, to really make that work commercially. And then in the medium to long term plan is to use these data to generate breeding values for facial eczema for the LIC bulls. We're probably looking at least 2 to 3 years out to be able to successfully do that. And genomics, we need a much larger data set. The sheep researchers used a data set of something like 16,000 sheep to develop their genomic prediction. We'll probably need something certainly in the thousands anyway to get a good handle on FE genomics, but that will be the most efficient way of identifying animals with FE resistance. So that's where we sit at the moment around facial eczema, and happy to deal with any questions, or if anybody wants to send in questions by email, that's fine by me too. So Esther, over to you.
If there's globally been anybody that's solved facial eczema through genetics.
The sheep industry in New Zealand is the probably the only example. Solved is not perhaps the right word either. You can't solve it, I don't think, through genetics. You can increase the threshold, you can increase resistance, but there are still some inherent risks with the toxin. I should have mentioned that the facial eczema is not unique to New Zealand, but it's certainly most prevalent here. There's a bit in Australia. Around Gippsland in particular, in dairy, and it's becoming an increasing issue is what I read. A little bit in South Africa, a little bit in Brazil, and last but by no means least, the Azores have a problem with facial eczema too.
Thank you, Steve. There's one more question just come in from Rosemary. Is there any indication of breed component to severity?
Yes, the work that Chris Morris and Neil Cullen did in the 90s would suggest that Jerseys were more susceptible.
Were there any indications why?
No.
So that's something to work on then, and that should come out when we have a bigger amount of data as well. There's another question just come in that really streaming in now. One from Catherine. Do you know if they were using zinc to control facial eczema in Europe, or is it not really an issue there?
It's not really an issue there. The zinc, as I understand it, is used in pigs in particular, pig industry, to control diarrhea in piglets. So it's really used as an antimicrobial.
I think the other point there is just, we are probably, New Zealand and Ireland are the biggest grazing populations in the dairy cattle population. So, in Europe, a lot of animals are being kept inside. So it's a very different environment, and they're struggling with very different diseases and issues around there. Another question there from Andrew Wallace. Is there any progress in breeding grass to be inhospitable to the fungus?
I don't know there's any work specifically going on to breed cultivars that are resistant, say amongst ryegrass. I'm not familiar with the literature on that, but I've certainly not heard of anything. But those different types of grasses, like kikuyu, like tall fescue, or crops, are probably the simplest route of getting rid of the fungus, at least during the summer months, or preventing ingestion of the toxin in the summer months.
Yeah, and correct me if I'm wrong, Steve, but I suppose a big issue with some of the crops and all the other things, potentially some annual ryegrasses as well. It's just the fact that you have to be reseeding it.
Yep.
So having a sustainable, high productive grass type will be hard.
True.
There's another question there just come in from Joe. What's the best way to find out about FE exposure in calves? Only blood tests?
I believe so. You could monitor spore counts in feces, which some vet clinics do offer that service. And certainly in mature cattle, something like 70,000 spores per gram of feces is the threshold at which you start worrying about the level of a toxin ingestion. But bloods would obviously give you gamma GT levels, GGT levels, which is potentially a little bit late in the piece to know that your animals are being exposed.
Yeah, that's great. Thanks, Steve.
Thank you very much for that, Steve and Esther. Extremely insightful presentation this afternoon. Thank you for your time this afternoon, folks. Thank you for joining us.